Okay, what we're doing here is we're going to set up the Darwin Seeds experiment. So what is the experiment? Why is it called Darwin Seeds? It's called Darwin Seeds because Charles Darwin did an experiment very similar to this and published a short little note or paper on it, and you're going to be doing the same thing. It has a little bit to do with plant germination, which is something you're also studying, and it has something to do with island biogeography and selection for characteristics and traits, and so a number of different things that are part of the course have some tie to this particular experiment. So what we're going to be doing essentially is creating seawater, putting it in some cups, and then we're going to get some plant seeds, which are provided in your kit. So you should have these little tubes like this, and they should have different species of plant seed in them. You're then going to put some of the seeds in the water, some of the seeds not in the water, and then you're going to let 2 days go by. And then what we're going to do is then remove the seeds from the water, and then the seeds that never went in the water, and we're going to set them up and see if they germinate or not. So essentially what we're going to be testing is are plant seeds that are exposed to salt water more likely or less likely to germinate, or equally as likely. It makes no difference whatsoever. So that's kind of the idea. And we're going to be using 3 different species of plants, so we have something to actually compare. So first off, we're going to set up the water, right? So you'd be better if you use like spring water than tap water, so there's no other chemicals in it. So what I'm going to do here is make up oceanic concentration salt water, but just using normal table salt. This is not chemical grade for lab grade. This is just from like a grocery store, sodium chloride salt. So you can just use this. Real oceanic seawater has, in addition to sodium chloride, magnesium chloride, calcium chloride, and a variety of other types of trace elements, but we're just going to be using the sodium chloride. You're going to make up 100 milliliters. So you don't have to graduate a container like this, just some kind of bigger container. And you're going to have your little cups. So I don't have the little cups here right now. I have something slightly bigger, but you'll just be using the little medicine cups that are in your kit for the experiment. To mix it up, you can mix it up in any container. So you're going to start with 100 milliliters. So how are you going to measure 100 milliliters for sure? These are included in your kit. It goes up to 50. Okay, so you're going to just pour water in here up to the 50 mil mark. I need 100, so I'm just going to have to do that twice. Okay, 100 milliliters. Now, oceanic seawater is 32 parts per thousand, so that would be 32 grams of sugar in a liter. But I just did 100 milliliters, so that's just 1/10. So I moved the decimal place, that's 3.2 grams. So you have a digital balance in your kit. So I turned it on. Put a weigh boat on it like this, then hit tare, so zero it. Make sure it says 0. Now you're going to measure 3.2 grams of just salt. So I'm going to do that. Doesn't have to be exact, just be very close. So oceanic seawater ranges, if you say 30 to even 35, but 32 is sort of the average. So take this, dump it in here, but the thing is you really need to mix it. Sometimes people forget that it has to dissolve, or it's not really the right concentration. So if you see salt on the bottom, you need to keep mixing it until it's all dissolved. Once it's all dissolved, just kind of you're going to again have the medicine cups, the smaller ones, but you're just going to kind of pour. The exact amount doesn't matter. This actually doesn't matter how much goes in these. You just kind of make it roughly equal between your containers. Okay. Now, you should have in these little tubes some seeds. All right, you should have 6 seeds of different species, and then another one we have a lot. There's actually 34 of the seeds. So what you're going to do is take 3 seeds, 1, 2, 3 of one species, put them in here, and then the other 3 that you have in that tube, just put them on the side. Keep them dry. They're not going to be exposed. Okay. Write down the time, the day when you start this. Okay. Then you're going to have the other species. Again, you'll have a little tube like this. I'm just pulling them from the original containers. Put 1, 2, 3 in there, and then again, the other 3 would go on the side. They wouldn't go in the container. So you repeat that for all of them. Okay, see, and then let it go for 2 days, 48 hours. Now, as it's getting to the time that you're going to pull them out, what's going to happen is you need to set up a little germination experiment. So what you're going to do is take a little bag, put a little piece of paper towel in it like this, which will serve a couple purposes. It'll hold the water, and it'll allow you to see the seeds, visualize them. I'm going to want you to wet this. Okay, so I'm modifying the germination experiment instructions, which the current ones I have posted. There's too much water in them. So what you're going to do is, I forgot my pipette. All right, so you're just going to put about 1 milliliter in here. I'm just going to spray it. I'll give you an update on the exact amount, but there shouldn't be a lot of water sitting on the bottom. You just really want this paper towel to be wet. That's all. You don't want a puddle of water on the bottom, because that could affect the experiment in a way that we don't want it to be. But we want to have moisture in here. So you just got kind of wet paper towels in here. Now, one of them is going to receive the dry seeds, the seeds that never went into the salt water. Ideally, when you position them, spread them out so that you can actually see them. Like this. And then you're going to need to label this dry seeds. Now they're wet, but they were initially dry for the 2 days. Then in the other one, you're going to fish out these seeds that were exposed to the salt water. So in this one, you're going to write salt water, and put those 3 in here. 1, 2, 3. Okay. So I have 3 seeds, and I'll have a bag labeled dry. They were never exposed to salt water. And the other, salt water. These are the ones exposed to salt water. Same species. You have 3 of each. And then you're just going to let them sit, and it'll take about 1 week. You really need to let it go 2 weeks, though, to be sure, because some of these species will germinate within 4 to 5 days, and some of them could take 8 to 11 days or so to germinate. So if after 2 weeks there's no germination, that means something bad happened, like the salt water killed the embryos inside the seed and they're never going to germinate. So if it goes more than 2 weeks, nothing's going to happen. But you really need to let it go up to 2 weeks. So the actual setting it up isn't going to take you very long to do, but you are going to have to wait 2 full weeks to get the results. So you need the experiment to get set up as early as possible, or you're not going to have enough time to complete it. And so you'll have bags like this for all the species. So you'd have 6 bags total, 2 for one species, saltwater dry, 2 for the other species, saltwater dry, 2 for the third species, saltwater dry. Okay, that's how this experiment will be set up. And then you're just going to be scoring them based on yes or no, they germinated, they did not germinate. So that just means the little embryos come out of the seed, you see a little plant starting to grow, or not. And that's the idea.